Hello, guys. Hope that you're good. Last time we discussed about necrosis, which is a type of cell death, which is caused by different pathological conditions. Today we are going to discuss another type of cell death, which is known as apoptosis or programmed cell death. If you look at the literal meaning of apoptosis, it means leaves falling off trees. And if you look at the details of this mechanism, there is a great deal of similarity with that particular, phenomena as well. We call it a programmed cell death because the programming for this particular, uh, you know, uh, cell death is there in the genomes of the cells. Or organisms are made up of different types of cells, but every cell type has got this programming. And this programming is uh, initiated and carried out with the help of different gene products, different genes encoding for different proteins. That's why we call it programmed cell death. So it's an encoded mechanism for cell death. The genome is just like a chip which has got this different information, different portals connecting with each other, different pathways are encoded, with the help of different genes. So it's a genetic program which is initiated in the cell when the cell feels that it no longer can survive in the face of, certain pathological conditions. The reasons for apoptosis are always either pathological or physiological, but while the reasons for necrosis are always pathological. We have discussed necrosis before that, and I hope that you would remember that. But it's a unique case here because this kind of cell death also occurs normally. So it's you can say a normal physiological mechanism of the body as well to die by this particular process because not only the cell, would die because the pathology is so severe or the disease is so severe, the infection is so severe that the cell no longer can withstand that and the cell decides to die. But there could be other reasons as well. Certain cells should be replaced with with newer type of cells. Certain cells required at particular developmental stage may not be required later in life. So the body has to get rid of such kind of cells. And that is a normal process which is going on inside our bodies, but we don't feel it. When you don't feel it, you should just, think about it that this kind of mechanism is something which is normal to the body. And the body does not create any inflammatory response. Otherwise, if there were inflammatory responses, then you would have felt it through the pain, or redness or swelling or some other cardinal signs of inflammation. But there is no inflammation in the case of a programmed cell death. So cells do not live forever. We understand this particular fact. It's very important for the students to keep these things in mind that cells are continuously replaced. They have a definite, lifespan, and after that, the cells are replaced with newer types of cells in case of different tissues. So there is a physical life of cells. For instance, if you look at the red blood cells in your blood, they would live for 120 days. So the cells produced today would live in my body would live for 120 days, and after that, these would be replaced with newer RBCs formed inside my body. Similarly, if you look at the white blood cells, they live, quite a short life, 13 to 20 days. Um, the hair follicle cells, which are one of the longest living cells in our body, they live for 3 to 5 years in our body. So after these they complete their lifespan, the cells are replaced with newer type of cells. How would these older cells be gotten rid of? So the mechanism for getting rid of these older type of cells which are no longer fully functional, or they are too old to perform their functions, the mechanism for that particular thing is here. It is apoptosis, or they are gotten rid of by the process which is already programmed there in the genome when we call it apoptosis. Similar is the case with the infected cell. In the case of infected cells, the infected cells mean that the reasons here is pathological. There is some kind of infection with virus, bacteria, fungi, or some kind of parasite. So these pathogens also can infect different cell types, and when they replicate inside, the cells, they would interfere with the cellular machinery in such a way that the cell would no longer be able to survive or perform its function normally. So in that case, the cell would decide that it's better to die rather than to live in this state. So such kind of cells can also die by programmed cell death, or the infected cells can also die by the program which is encoded in, side the genome of that particular cell. So this kind of, um, that you can call it a suicide rather. So apoptosis or programmed cell death is suicide of the cell because the cell is active here. The cell is taking part in this particular process in decision making that whether it should survive or it should die. So if it feels that survival is difficult, in that case the cell would decide to die. Certain cells would be defective, and defective cells are also removed out of your system continuously, and they are formed in almost every individual. Certain cells would have certain defects. For instance, the lymphocytes, when the lymphocytes are formed, sometimes the lymphocytes, for instance, the T cells, they would have defective T cell receptor, and uh, that T cell receptor would be defective in such a way that the TCR, or the T cell receptor, which is formed on the surface of them, would react with own, immune cells, or immune cells of that particular person, which would cause different kind of autoimmune conditions. In that case, it is very detrimental to that particular individual. That's why, the the cell would decide that in this kind of situation it's better to die, and the program for cell death or apoptosis will be initiated in defective cells as well. So sometimes the cells would be produced in excess, uh, and these excessive cells, would be removed out of the system by this program cell death or apoptosis. For instance, in the bone marrow house, the cells that are formed, extra cells, are not allowed to join the blood, and the extra cells which are formed there, be the RBCs, red blood cells, or white blood cells, or platelets. These are the cells that are in excess. They are, they undergo this process of apoptosis in the bone marrow house, and they are not let to join the bloodstream. Otherwise, they would cause a lot of problem for that particular individual. Similarly, extra cells in the gastrointestinal tract, they also undergo apoptosis, and you would find lots of different types of GIT cells in the stools of individuals. Sometimes these stools are also used for genetic testing in different individuals. That because the cells, the apoptotic cells, they are usually present in the stools of individuals. Certain cells would be hormone dependent cells, and the hormone dependent cells, um, uh, at certain particular developmental stage, some cells may be required, and at other stages, these cells may not be required by the body. So in that case, too, um, what would happen when the hormone is dropped at certain point of time, then the cells which were dependent on that particular hormone to survive would also undergo apoptosis, and these cells would no longer remain there. It happens in the case of menstruation. You can see the endometrium there is continuously sloughed up after a month in females. In other cases, you would have seen during embryonic development, for instance, when the vessels are formed, these vessels are usually, these are tubes. The blood vessels, for instance, or other vessels, they're filled with, tissue, and that tissue has to be degenerated, or that has to be gotten rid of when the vessels are getting ready for, circulation. So that degeneration of internal luminal tissue in order to make a lumen or a narrow channel in the vessels, that also takes place by this process of apoptosis. And similarly, the web in the in case of embryo, for instance, the human fetus, when it is formed initially, there is a web in between the fingers of that fetus, but that the tissue in between the digits is degenerated with the help of apoptosis. So these are normal physiological reasons which are, you know, uh, actually causing this programmed cell death, or which initiate this process of programmed cell death in our body. And the reasons can also be pathological. The pathological reasons that we have studied in the cases of necrosis, which include these infections and chemical injury and other types of injuries. These also can cause apoptosis, and uh, apoptosis can also be caused by this particular physiological reasons. So you just remember that apoptosis is not associated with inflammatory response or inflammation. That happens in the case of necrosis, but not apoptosis. And the second thing is, the integrity of the membrane, the plasma membrane, is not lost in apoptosis. For instance, if this is a normal cell undergoing apoptosis, what happens when this program is initiated, then the cell shrinks. The chromatin inside the cell, it condenses. So that is the first step which occurs, the shrinkage or the condensation of the chromatin occurs there. And then there is the blibbing. The membrane starts blibbing, and the organelles are disintegrated. If you look at the structure of the plasma membrane, you would have seen the orientation of the phospholipids and the glycerol and fatty acids there. So the fatty acid stays are always inwards. It's like this. But in some cases, in an apoptotic cells, uh, uh, the membrane, the structure of the membrane is changed in such a way that the fatty acid tails are directed outwards. In some of the fatty acid tails are directed outwards, and that actually is a signal for these cells, the macrophages, to send such kind of cells which are undergoing apoptosis, go there and attack them, grab them, and get rid of them. Okay, so they are not taking on whole cells. Rather than this blibbing which is taking place here, you can see the material has condensed, and the blibbing is taking place, and this increasing blibbing which is taking place here is converting this entire cells into small bags. So these small bags, which are surrounded by this, structurally different plasma membrane, are taken up or sensed by these, macrophages, and they would just, engulf these blebs and got rid of the cell. So the cell is gone, and it would be replaced by new cells. And there is no inflammation. Why there is no inflammation? Because you can see that the membrane is almost intact in this entire process. Nothing is coming out of the cell. The content of the cells condense. The enzymes which are inside the cells, the enzymes like these hydrolytic enzymes, lysosomal enzymes, peroxisomal enzymes, all these enzymes are intact there, and they are also packaged in these packs, and they would just be engulfed by the macrophages, and in such a way, the cell material would not go in the open in order to destroy other cells or the extracellular matrix or to initiate the process of inflammation. So that is the difference between necrosis and apoptosis as well. In necrosis, the membrane is damaged and everything just comes out of the cell. The lysosomal enzymes and other bacterial enzymes, they would just initiate a process of inflammation. So where there is necrosis, there is inflammatory zone around that particular area, and where there is apoptosis, there is no inflammatory zone. It's a normal response of the body which can happen. Now there are different factors which can cause apoptosis. We call them extrinsic factors or intrinsic factors. These extrinsic factors are coming up from the outer world. The signal is coming from outside the cell, while intrinsic factors are inside the cell. So first we would discuss about the extrinsic factors, that what are the extrinsic factors which can cause apoptosis. And extrinsic factors, you can say that there are receptors present in the membrane, because the membrane, the phospholipid membrane, is embedded with many different types of receptor proteins. There is a particular receptor protein which are known as the death receptors. We call them the death receptors. If the death receptor found its suitable ligand, which is called the death ligand, that death ligand can uh, that ligand can actually lead to activation, dimerization of these receptors on the surface of the membrane, plasma membrane. When the ligand binds them, these are accumulated or they dimerize. So that is the active form of this particular receptor, and then this dimerization actually is leading to send signals internally. Okay, so what is the death ligand? First we discuss this. The death ligand, the tumor necrosis factor alpha, or the FSL, fast associated signal ligand. These are the death ligands. They are special kind of ligands which can activate this particular receptors. And these are the death receptors. There are different names given to the death receptors. For instance, FAS are the first apoptotic signals, or you can say TNFR1. There are different names given to it. Some even call it APO1, APO1, or tumor necrosis factor, RSF6, or CD45. There are different names for this particular for these receptors. Um, APO1, or apoptotic antigen 1, that is also the name given to this particular receptor. So these receptors, when they are bound to the ligands, or you can also call them the we also call them fast ligand receptors. These are the fast receptors. These are the fast ligands. So when the ligands they would bind to these receptors, the receptors are activated. Now, these receptors are internally are attached with adaptor proteins, and these adaptor proteins have a domain, which is actually a dead domain. You know, this dead domain is, you can say, this particular domain, dead domain, it is known as DED. Or you can say that when this adapter protein is activated, the adapter protein would, so these adapter proteins, they have a dead domain, which is known as the fast associated dead domain, or FADD. This is the fast associated dead domain. And then this fast associated dead domain, you know, this fast associated dead domain, along with these different kind of proteins, would make a complex which is known as DISC. Or this DISC actually is the death-inducing, signal complex receptors. So the DISC actually is now acting upon K spaces or the enzymes which are going to bring about degradation of the internal cytoskeleton and other chromatin material. So there is a dead domain on the internal side of these receptors, the death receptors as well. And that internal dead domain activates the adapter proteins, which has their own death, domains, which are known as FADD, fast associated dead domains. Then this complex, this death inducing, signaling complexes of receptors. This, it acts upon pro-caspase. The enzymes, caspases are enzymes, they are going to bring about the degradation of the internal cytoskeleton and other material. There are different caspases. Some are activated early during apoptosis, and other caspases are activated late. So in early caspases that are activated early are called procaspases. We call them procaspases. It's a family of proteolytic enzymes inside the cell. So the caspases would be activated early during this particular process are called the initiator caspases. For instance, caspase 8 is an initiator caspase here. And the caspases which would be activated late during the process are called uh, executioner caspases. So initiator caspases would initiate the process of, this process of apoptosis or this process of degradation. But initial caspases are not going to degrade the cytoskeleton, etc. structure. Initiator caspases are going to activate other caspases, which are known as executioner caspases, and those executioner caspases are going to do the job. They have to bring about this degradation of the cytoskeletal structures and the chromatin material inside the cell. So you have to differentiate between these two different types of receptors, the enzymes. So these enzymes are, when they are present normally inside the cells, they are not activated. We call them proenzyme. For instance, pro-caspase 8. This enzyme here, pro-caspase 8, with these domains, is not an active form of enzyme. So what is the DISC complex doing? This complex, which has been found by the assembly of the fast ligand with fast receptors and adaptor proteins, This particular complex or the disk is going to activate procaspase 8. So the FADD or the fast-associated dead domain uh, of this complex is going to activate procaspase 8 now. And the procaspase 8, it would rotate with the procaspase 8 in such a way that the procaspase 8 would just open up and start cutting, doing its job. And this procaspase 8, this autocatalytic activation when the caspase 8 is activated, this initiator caspase will then go on and, it will just cut other caspases or other, enzymes, activate other enzymes, other caspases, which are known as late caspases. We call them caspase 3, caspase 6, and caspase 7, for instance. So these caspases would do different kinds of jobs. So for instance, these caspases would start, caspase 3, 6, and 7 would start cutting the cytoskeletal filaments, actins, and other filaments which are there, and they would the DNase it would also be activated as a result of the action of these enzymes, which is going in a sequence, one activating other enzymes, and there's a group of enzymes which are now active in cutting different structures. So the chromatin would also be cut at different places. The blebs formation would ensue as the cytoplasm is shrinking, and the internal material are cut with the help of these enzymes. So the blebs would just go inwards and inwards, and the cells would be packed in small bags or packages. Small packages would be made, just as I show you here in this slide, that the cell is packaged in the form of small bags, and then these bags are taken up by the macrophages. How the macrophages sends to take up these bags, uh, as I told you that the plasma membrane, integrity of the plasma membrane is compromised, and the fatty acids tails also sometimes would go outwards rather than pointing inwards, which is a signal for the macrophages to sense and go there and engulf that particular blip or that particular part bag which is filled with the cellular material. So in this way, the entire cell will be gone and it would be replaced by other types of cells. So this is the extrinsic. These are the extrinsic factors or the mechanisms that extrinsic pathway of apoptosis, the factors which are responsible for initiating this extrinsic pathway of apoptosis, these fast ligands which are activating different genes. So for all these genes, for these receptors, the adapter proteins and caspases, there are genes in the genome. So all these are encoded in the genome. And these are activated once they get the signal through the fast ligand, or FASL, which binds to the receptor. So these receptors are particularly made for initiation of the death of the cell. So the cell is actively involved here because the signal which is received here is received by the cell, and the cell is transmitting this information inside the cell in order to activate this machinery, these caspases, in order to bring about its own death. That's why we call it apoptosis. So that's all for today. Maybe tomorrow we will discuss about intrinsic pathway of apoptosis as well. Thank you very much for watching.